biology and the cell culturing, right? So this part, what I'll do is probably after once we finish a portion, that is, whatever the lessons are there, all the topics in the next 10 days or the next 8 days, I will just, cover this up so that you will not be missing on the portion. Because we have certain things such as model organisms and the tools that are there in the cell biology. The microscopy part, I will be taking it right now. But otherwise, you have other things such as metal shadowing, centrifugation, and all of that, right? So this part will be dealt with a little while later. I hope this is okay with you. Just like, put in a message for me. And today, I will take a unit 2, which is basically an overview of the cell. Wait, just a second. Yeah. Methods in cell biology, right? So this has your basic, what we are going to study in this is the cell structure and cell function we have already seen. So in this, we are going to see about the application and the principles. As such, how are we going to see the cell structure? How are we going to see the fractionation of cells? And then apart from that, we are going to just have a look at the microscopy and what exactly are electron microscopes that are present. You have your transmission and scanning electron microscope, and at the same time you have the fractionation of cells, right? So what exactly is flow cytometry? How do you do cell disruption? What are the methods of cell disruption? Then how are you going to separate the different organelles, like using your centrifugation? And then what are the antibodies specific to different kind of organelles for the purification purposes, that is. And then we are going to see what exactly is tracer technique, and then what is autoradiography, and then we'll just have a look at the summary. So let me just start with the basics of microscopy. I'll just share my screen with you. I have a couple of so yesterday's topic, microscopy, we have different types of microscopes, right? So I will just start with them, in fact. So today, yesterday's topic also will be completed, at least the microscopy part. So I will just kind of share the screen.
Good morning, ma'am. Can you tell me page number, ma'am?
I'm not exactly going by the page number, so just don't bother with it right now.
Okay. Lessons. What is the lesson, ma'am?
Okay, what you can do is open your unit 1 microscopy part. At least the basics of microscopes, I will be just be dealing with that. And along with that, you have your bright field, dark field. So today
Oh,
it will be microscopy.
okay.
I would rather you just listen to the lesson which is being taught right now, and then you can actually go through that once the lesson is done. Try to share my screen. Shit. So when it comes to microscopy, so
Yes,
basic,
ma'am.
yeah,
Audible. It's
yeah.
audible.
So when it comes to basic microscopy, what we are just like looking at is it is a magnifying glass. The simplest way or definition of how we are going to look at microscope. So it is an instrument that is going to magnify any object that you keep. You don't have to keep only cells. Any object that can be fit in, under the under the eye lens, and then you see it is going to you're going to get a magnified image. So it is that instrument that magnifies the size of the object and interpretation of the magnified images. Are you able to see the screen?
Yes, ma'am. It's
Yes, ma'am.
visible.
Yes,
Okay.
ma'am.
So microscopy is the technology that is going to make small things visible to the human eye, and you're going to magnify it to n number of times. It can be like 1 lakh times in the case of scanning or transmission electron microscope. Sometimes it is only magnified to 10 times, and 1000 times, 100 times, 45 times. So you have 10x, 45x, 100x in your basic lab microscope, which is the compound microscope. When it comes to bright field, dark field microscopes and your scanning and transmission, you're going to magnify it, or you can see the image in a very clear manner up to 1 lakh times magnified. So it is very apt to look at, and it is very useful to look at the small cells that are present. So who was the first person who actually discovered the fact that you can see small things is Anton von Leeuwenhoek in the year 1676. So he had in like I wouldn't say discovered. I would say he invented using a small biconvex lens. He managed to, once he got the hang of optics, so he put a small single biconvex lens, and then he could see different cells. He used his pus cells from the teeth. He just like took that. He saw onion cells, all of that together under the microscope. Then Robert Hooke pointed the cell, he built microscopes with two lenses, and then he was the first person to invent the compound microscope. And then you have Zacharias Janssen and Joseph Lister. All of them had made advances in the different kinds of microscopes that are present. So it is important today. All of us know for basic reasons as well. So you're going to see any kind of, you put in a slide, you're going to see it under the microscope and see that unit is magnified. And by giving the resolution, that is the with the help of clearing or like properly adjusting the eyepiece, you're going to get a proper image. So all the organisms or substances, bacteria, viruses also are going to be magnified, and you can see them with your eyes. So it is a major tool for any kind of microbiologists and biotechnologists. So what are the factors that are present in the microscopes? If you just see, you have the magnification, you have contrast, that is how much big you can make the image. And then you have in different light, how are you going to see according to the different intensity of the light, how is the image being visible, what is the resolution power. And then you have different kinds of microscopes that are present. So I'll come to that again in a minute. I want to deal, yeah, sure. Today I will just talk about the magnification. So what is it? Magnification is the power of enlargement of an image present in an object. So what is the size of the image? To the size of the object that you're seeing basically. So magnification is the image distance that is present by the object distance. So you can see the image with your eyes from the eye lens, and then the object distance, how far it is. So there are three types of lenses that are present. Usually low power. This is in the basic compound microscope. 10 times magnification. High power is 45 times, and you use oil. Cedarwood oil is used basically, so it is going to be like 100 times magnification of the image. So if the image is 1 mm, you're going to see it as 100 mm or 1 centimeter, what do you call it, 10 centimeters. For example, I'm telling you, please do not take it to the T. And contrast is in different light intensity, how is the object being seen? So it should have a you should be able to look at the image and tell it apart from its background. So that is its contrast power. And when it comes to the resolution power in the microscope, what is, how clearly are you able to see two different objects in the microscope? So that is the resolving power present in the microscope as such. So what is it that you're going to see? If you're going to see two organelles, say for example, your mitochondria and Golgi complex, so you should be able to distinguish between both of them. So that is the resolution power in the microscope. In this, it is again determined by three factors, which is what is the lens size that is present, one thing, and the amount of light that is being passed through. So the wavelength of the light that is there, that is going to hit the specimen. So this gives you the proper resolution again, and the refractive index of the material. So what is going to happen is there any kind of see the basic example is refractive index is it is going to be different for oil and for air. If that, so that is the refractive index of the material between the objective lens and the specimen. Now we have different types of microscopy. I would like to share one more slide with you so that you'll be able to understand. So introduction, we have seen what is an instrument. microscopy, and then historical background also we have seen. Zacharias Janssen has developed the first microscope, and Huygens, I don't want to even know what are the variables. So when it comes to magnification, this gives you a simpler example. In fact, what is magnification? Degree of enlargement. So how many times the object is going to be multiplied or becoming bigger? What does it depend upon? The tube length, focal length of the objective, and how big, or what is the magnifying power of the eyepiece. So complete magnification is total magnification is magnification of the eyepiece into magnification of the objective. Resolution, like I already told you, depends upon different factors. So how closely you are able to differentiate between the different objects that you're seeing. And limit is again how much you can separate and how much you cannot see. So less, lesser limit of resolution is this number has been seen. It is based on the different kinds of eyepiece distance and the distance between the slide share. So into wavelength and the numerical aperture. What exactly is numerical aperture? Again, you can see it is the refractive index. So all this is basic maths formula that you know through the if you know the microscope in detail, then you will get to know of it. And then definition is the capacity of an objective to render the outline of the image of an object clear and distinct. As such, what is going to happen is how clearly are you going to see the object? Aberration is the difference, the minute or the actual difference between where exactly the image is and where exactly is the the actual image is present, and how much are you able to see that minute difference. So you can see that you have two different types of aberrations over here. One is the spherical aberration and this is the chromatic aberration. So you can see that it is the focus of axial axis rays that are coming, marginal rays. So this I would have to start with the proper microscope and its mechanics for that, which I will deal with, like I told you, in later class when we get the time. Because right now I want to get into unit 2, which is like your bright field, dark field, phase contrast, microscopy, all of this. So I'm just going through this so that you'll be able to understand the the phase contrast and the bright field microscope later. So when it comes to spherical aberration, you're going to see that it is going to be in this manner, wherein you have the difference over here. You're supposed to be able to see the image over here, focus of the marginal rays. So you have the rays of the eyepiece coming over, and you'll have to be able to see the image over here. Instead, you're going to see the image over here. And at the same time, axial rays also, you should be able to see over here. Instead, you're going to see the image over here. So this difference is known as your aberration. And when it comes to your chromatic aberration, the focus of all the image you're going to see in the axial, what do you call it, the marginal rays, you will be able to see over here. Whereas your basic axial rays, you'll be able to see. So this difference is the chromatic aberration as such. Now we have something called as the working distance, which is the distance of the front. So wherever the lens, this is the slide of the specimen that is present. This is the objective. This should be the distance. And this is calculated with an angle which is known as theta. So this is the theta that you're going to get. So when you're talking about contrast, what is it that you're seeing? The intensity of the difference between the two different, the images. So difference in the intensity between the two objects or between the object and the background. So you're able to see this is contrast. This is the background. This is the cells that you're able to identify and distinguish between them. So what is it that we are looking at in a microscope? We are going to see the properties as such, yes. The slides are not visible. Or you should have told me a little while earlier, no? Oh, is it visible right now?
Yeah, ma'am.
Okay, sure. So this is what I was talking about. Numerical aperture is the theta. Theta is the objective lens. See the specimen slide, and this is the objective lens that is going to be here. So this angle you see, this whole thing is theta. And since between B and D, angle is only half of it, you call it as theta by 2. And N is the refractive index that is present. Okay, when we, and like I told you a little while earlier about spherical and chromatic aberration. So you see, chromatic aberration means basically your axial rays and your the marginal rays that are present, you're able to see in a different way. So this particular distance or difference that you're going to see is known as your chromatic aberration. Spherical aberration is, again, your lens is over here. You're going to see that the axial rays and the marginal rays, wherever they meet, there's a difference over here again. So this is your spherical aberration. So I was talking about this till now. Now you see, this is the objective. This is the slide with the specimen on it. And like I told you, this is the theta or the angle that you see. Now, contrast, like I told you, you're able to see the images over here, right? So when you're able to distinguish the images between the background, that is the contrast we are talking about. So for the microscope, we have certain properties that have to be taken into consideration. That is your magnification, the numerical aperture, and how much of focal length. When you increase and decrease the focal, you can do that. And the working distance and the resolution power. So it differs according to different kinds of magnification that is there for basic simple compound microscopes that are present. So in a compound microscope, you can see this is the ocular or the eyepiece. This is the body. This is the nose piece. This is the objective lens. So this is different types. You must have already dealt with microscope so far. So I'm assuming that you know that there are magnification lenses, different ones, 10x, 45x, 100x. Some of them have even 30x. So according to that, you can just adjust the eyepiece. This is the mechanical stage where you put in the slide and you have the adjustment knob over here, coarse and fine adjustment knob. This is the stage adjustment. And then you can control the light intensity over here. And then you have the light source also available over here. You have a magnifying glass and I'm sorry, it's not a magnifying glass. It is a condenser and you have a diaphragm control. So you can just adjust that and you can adjust the intensity control. And this is the basic compound light microscope. So what is it that we are seeing over here? We have to measure the size of the microscopic objects in a compound microscope. How are we going to use that? We are going to use a micrometer, which is the stage micrometer, and the eyepiece. And you have to do calibration, that is how correct it is. So for that, for a stage microscope, you have the calibration lines or the graduation. They are separated by 0.01 millimeters, that is they are 10 micrometers apart. So you have the micrometer over here, you see, and this has the retaining ring, so it is being inserted into the base. Okay, and then this is the stage micrometer. You're going to put in the slide over here, position it, and then you're going to adjust the light source underneath so that there is light being focused over here. And then after the completed calibration, you're going to put the slide in for measurement of the object that you're going to measure. So it is achieved by, you're going to superimpose both the scales, the micrometer scales and the eyepiece scales together, and then get in the size. So this is the stage micrometer and this is the ocular micrometer. You're going to just put them together and then you're going to be able to measure the size of the cell or cell organelle that you're going to see. So they have just given how to do that. Basically, each graduation is around 0.0014 meters or 1.4 microns. Then it is going to appear in this manner and there is a spacing that is present. And then once this is done, you're going to put them in the, underneath the lens basically. Then you're going to see it's going to appear like this. So it's around 10 micrometers apart. And on the basis of this, you can calibrate. See over here, there's a cell or a cell organelle. If you want to check the size of the RBC or size of the yeast cell with the bud, You can see they have already shown you an example. So on this, you can tell the length of the yeast cell and the bud that is present. So different types of microscopes are there, light microscopes and electron microscopes, which is the advanced one. So you have the bright field, dark field, phase contrast and fluorescence microscopes. And we have electron microscopes, wherein electron beams are passed through. That is a different story altogether. We will come to it in some time. We will still have a look at it. So what does a bright field microscope do? It's going to produce an image against a, it's going to have a dark image against a bright background. So you have different objective lens and there are two types again. So when it comes to that, you have the simple microscope. It's only a basic magnifying glass. Compound microscope, again, like I told you, 10x, 45x, 100x. So you have a series of lenses for magnification. Now light passes through the specimen and onto that objective lens. And in case you're going to use 100x resolution, you're going to have the cedarwood oil being applied between the slide and the glass that is provided. This will increase the resolution. So with that, you have a resolution of up to only 200 nanometers. So when it comes to that, you have a glass cover slip. What is the oil immersion or oil which is going to be useful for, so you have without oil immersion, this is the light source. You have the glass cover slip over here. This is the oil that is being used over here. So what is going to happen is, this is the lens, right? Refracted lens. You have the axial rays which are actually being lost. But with immersion oil, what is happening is all the rays are concentrated on the objective and the lens, that is. So because of which you're able to see the image and the resolution is going to increase. That is why you use oil over here for 100 times magnification. So when it comes to the optics of the compound microscope, final image is where you're going to see over here. This is the eyepiece, projector lens. So this is where we have our chromatic and spherical aberration coming into place. So the difference between the eye of where we are going to see the image, where the actual image is formed. So this is the light piece. What is the advantage of a simple and a compound microscope or the bright field microscope? Used to see live or stained cells. It's a very simple setup. All the labs have this. But the disadvantage is that it is basically, you have, what do you call it? You don't get a very good fine image that you can see. And if you stain, it can actually destroy some of the cells that are present. And resolution is only up to 200 nanometers. So, bright field microscopy, you can just see the gram positive, gram negative bacilli. We have already done this earlier in our undergrad. So, when it comes to the dark field microscope, you're going to get a bright object against a dark background. Optical system is going to be going to be much more better as compared to the unstained bodies that are present. And then the specimen is going to be bright and the background is going to be dark. So what is it happening over here? So basic diagram, you can see you have a dark ground condenser, you have a high intensity lamp and a funnel or an object over, stop basically. So this is the objective, the lens. This is where you're going to see your final image, right? So dark field stop is present. You have the condenser lens. This is the stage and the objective lens. So dark background, you're going to see the light or bright, whatever specimen that you're going to see. What is it useful for? Different kinds of bacteria and different kinds of microorganisms especially. So you have Treponema, Leptospira, all of these endospores of the bacteria that can be used to see using the dark ground microscopy. Advantages, again, simple setup. You're going to see, unstained tissue is going to be present and against a dark background you can see, and you can see the live cells, so you do not have to stain. So staining actually has this disadvantages, but then you have to have a the disadvantage is that delicate samples, if you put such high intensity of light, it is going to get damaged. So the specimen, because we are looking at microorganisms, So such high intensity of light is actually going to cause some amount of damage. So we'll just have a look at phase contrast microscopy. Uh, just give me a moment. Right. I will have to actually go back to the earlier slide. You will like it. I feel that is a better one. So I'll just like put it in. So far we have seen the bright field micro. Are you able to see the image now? It says microscopy, the yellow slide, right?
Yeah.
One of you just put in a message.
Yeah, ma'am, it's clear. It's visible.
I will share both of these today in the group. Ask Nana to take it. I'm sorry about that. So yeah, we were talking about the dark field microscope. You're able to see now? So the background is
Yes,
going to
ma'am.
remain dark. Only the
Yes, ma'am.
objects are going to be illuminated. We have seen this. And there's a special kind of condenser lens which is actually going to concentrate all the light rays so that, you're going to get a better image as such. I'm trying to see as to how to remove the clipboard as of now. Right. So what will happen is this disc is going to block the light. So the rays are going to scatter like it changes the light, bounces off the small object that is present, and then you have a condenser and it has a dark field stop over here. See, you see this opaque disc. So because of this, what is going to happen is your light rays are going to get intensified, and then it enters the dark objective directly. Then again the light beam is going to be redirected so that the specimen, the light beam goes onto the specimen, but it is not going to go onto the objective lens. So only the light rays are going to enter the objective lens. They're going to, reach the eyes, and because of which your specimens appear dark, bright on against the dark microscopic field. So microorganisms are seen, motile live cells are seen in this, microscope, or like the dark field microscope. When it comes to fluorescence microscopy, you're going to see that it is what do you call it? Again differs from both bright field and the dark field microscopes. What is going to happen? Because you have a mercury vapor arc lamp, or you're going to use a halogen that is present instead of the incandescent lamp. So what is happening in the fluorescent microscopy? You have a fluorescent antibody. We know that our defense system has antibodies. So all these antibodies are your natural defense molecules. They're going to be produced by the immunoglobulins, for example. And then many animals, this is going to actually identify and destroy the antigens that are present. So when you're using this fluorescence microscopy, you have different types of fluorescence that is present. So we have a light source over here. That particular antibody is going to go attach itself to, see, basic principle of antigen antibody is antigen is a foreign substance. It can be anything, self, non-self. And the antibody will go identify it, and then it is going to attach itself to the antigen. So same principle is going to be applied when it comes to the fluorescence microscopy, wherein the cells are antigens and the antibodies that you're going to add, which is attached with the light source. So, with the principle of dyes, that is going to act as antigen antibody, and you're going to see a very beautiful colored image of the antigen, which is the specimen that you're looking at. So you have types again, autofluorescence. So you have a blue-green light that is going to come. Secondary fluorescence, you have the Congo red, eosin, they are the red dyes. And induced fluorescence, again depends. So what is it that we are going to see over here? Right. This is the light source. You can see this is a mercury arc lamp. You can use a UV light also, and you have to have a shorter wavelength over here. And heat filter. We want the light, but we do not want the heat over here because heat will destroy the organisms as such. So it will not help our purpose. And then you have the excitor filter. So we all know that light has different kinds of wavelengths present in it. So we want only one kind of wavelength that to pass through, and the rest of it should be should not pass through. So you have an excitor filter. And then you have a diachronic mirror. What is it going to do? It is going to, you have one excitor filter over here, so that wavelength is going to come. So now this wavelength light is going to be divided by the diachronic mirror. What is it going to happen? It will reflect light of one wavelength because already the filtered part only one is is coming out, and it is going to transmit the other. So, and then once this is done, you have the emission filter over here. And then this is the ocular and this is the detector. So the condenser, we again have a condenser over here. I'm sorry, I missed it. So in this, you're going to get a dark background and the fluorescent object is going to be there. So this is the specimen and this is the objective. So, and you have a barrier filter, like I told you, which is going to remove the excited wave. So what are you going to see in this fluorescence microscopy? You're going to detect proteins, and that is, you're going to stain the proteins and then detect it. And then you can see the chromosome branding. So all the bands in the chromosome, when you do cytoscopy, what do you call it, for seeing the different kinds of bands in the chromosome, where are the gene and the loci located. So you're going to use this fluorescence microscopy. And then fluorescent antibody technique or the immunofluorescence. All of this can be seen through your fluorescence microscope. Phase contrast is again rays of light. They are moving at different speeds through materials, because of which different refractive indices are there. And then you're going to see different kinds of images. So it amplifies this difference in the refractive index of the cell. So what is going to happen is, in different environments, that is, you're going to use aqueous environment, and then this is going to be converted. So in this, you have different condensers. Basic use of a condenser, it promotes the, or it helps in passing, or it intensifies or strengthens all the light beams and lets it pass on to the objective. So in the phase contrast microscopes, you have special condensers, you have special objectives, because of which you will be able to see the contrast between the different components in the cell. And that is because all the component cell organelles, for example, they are not going to have the same density. So if at all you want to view Golgi complex and mitochondria, you will use a phase contrast microscope because they have different densities as such. So you can study the living organisms and the living cell, to convert the invisible small phase changes. And this is that is present in the cell component into visible intensity change. So what are the parts in the phase contrast microscope? You can see over here. This is the eye. You're going to see from here. This is the ocular lens. You have an objective lens. You have a condenser lens. So you have three different types of lenses. Then you have a diffraction plate over here. Till here, from the objective lens, you have the rays coming over. See, finally, this is the light. You have the specimen present over here. You have a condenser light. So the rays, axial rays, are going to go through this and then on to the specimen. So far, you have one particular light going on to the specimen. And from here, you have refracted or the diffracted light that is going to go onto the objective lens. From here, you have another diffraction plate. In this, you are going to see the rays coming over, and then through the ocular lens, you're going to see from from the eye finally. So when the light rays are passed through the cell organelle, they're transmitted at a velocity inversely proportional to the refractive index of the cell organelle. So cell organelles have different refractive indices, because of which this particular point is being exploited in the phase contrast microscopy. Now the light rays that are emerging, they show the variable phase changes. So with this principle, you're going to see that you are able to distinguish between both your or cell organelles. So whenever there is this invisible phase that is present, it is going to again change, reconvert into visible intensity by a phase contrast microscopy. I hope this is clear. So what is it used? Where you're going to see that in case there is more refractive index, then the thickness will also be more. And then the change in the phase is also going to be more. And because of this, the cells and the components with the difference in the phase change value, that is almost like, 1/4 of the light that is being seen. This phase change is usually not being seen to the human eye. So you need one more lens to convert it so that we are able to make out the difference. So that is the principle behind the phase contrast microscope. Again, it is a specially designed light microscope and it has an annular diaphragm and an annular phase plate fitted into it. So you just can read. In the phase contrast microscopy, you're unable to increase the contrast between the transparent cells. So if you want to see the density, like I told you, you just have to change the, you have to use, see through the different lenses and they will help in changing or making out the difference and then you'll be able to see. So the light rays pass through the annular group of the annular diaphragm and these rays are focused onto that object and then two rays are emerged out. One is the refracted ray that undergoes a phase change and other is the central ray which does not undergo any phase change. You can just see here. So refracted rays are coming out and the central rays. These central rays do not have any phase change. So you have a diffraction plate over here. So finally, all these merge together, the refracted rays and the the central rays that are present. And then finally, you're going to see the lens with the differences in the, what do you call it, the between the organelles you're able to find. So again, in the phase contrast microscopy, what is going to happen is the refracted rays are banned due to the refraction in the density and then the refractive index within the specimen and then they get refracted about 1/4 of the wavelength. So you're going to use a phase plate for it, image formation. That is by putting the this image is formed by putting the subtractive superposition of the central and the diffractive rays and the object is going to appear dark against a light background and this is known as a dark positive contrast. So what is the advantage of this? We are going to see live cells. You don't need staining of it and even highly transparent. So you do not have to stain the cells. You can see the highly transparent material because you're going to see the difference in the densities as such. And then even the intracellular components can be observed such as the endospores. In the bacterial cells, if at all there are any kind of endospores present, you can see that. Now, when it comes to electron microscope, the principle is again very much different. You have electrons, they are going to illuminate the objects. And this is going to have a very great magnification. So this is, it can achieve a very high resolution. It uses electrons of the shorter wavelength. And then you can get to see that when the electromagnetic lens is going to focus a beam of electrons onto the specimen, you're going to get 10,000 plus, 1 lakh plus magnification as well. So you have different types over here. When it comes to the properties of the electron beam, you're going to see that the low energy electrons are emitted. They are known as the secondary electrons. useful for image formation, especially in scanning. You have two types of electron microscopes. One is scanning electron microscope and the transmission electron microscope. So whenever they are emitted out using electric current that is passed through a tungsten filament, you have a wavelength of the electron that is again based on the electric current voltage. So it is calculated by your de Broglie formula as such, and it is calculated and it is going to be in angstroms. So what is the principle of, like I told you, there are two types of electron microscopes, scanning and transmission. In transmission electron microscope, you're going to get a higher magnification, higher resolution. Wavelength is around 1 lakh times shorter than that of the light rays that are present. So you're going to get an excellent image. Then image is produced on fluorescence on the photographic plate. Instead of mounting, it is held in a proper way as such. I'll just right. So the total setup has to be kept under a vacuum evacuated column, and then you're going to so as to prevent the random collisions of that electron beam with the dust particles, so that there is no deviation from the specimen. So how is it going to be constructed? You're going to have a tungsten filament over here. You have a cathode and an anode that is going to be present. This is the first set of electric coil that is present, condenser. So hot tungsten filament, source of electrons. Electromagnetic lens that is present corresponds to the condenser, objective lens, and the ocular lens. Then microscope column is present. You have a fluorescence screen and a vacuum pump. And you have different transformers which are going to give you the set voltage, and it is going to be heat producing. So you need to have a water cooling system as well. Okay. So when it comes to the working basic, what is going to happen? The electron gun, which is a tungsten filament, it generates the electron beam, and then the electrons are that are present. They collide, or they, this in the form of this collimated beam, they pass through the condenser coil, fall onto the object. Scattering of the electrons is going to take place, transmission of it through to the object is going to take place, and then from the objective coil it will magnify the image of the object. The projected coil finally magnifies the image. Final image is going to be present in a fluorescent screen connected to your computer. So that is one thing. Magnification is almost like 1,60,000 times to 10 lakh times. Where is it applied? Everywhere. Viruses, internal structures of all the organisms can be seen through this. When it comes to scanning electron microscopy, you have three-dimensional picture that is going to get. So the whole mitochondria, for example, or the whole Golgi complex can be seen through the scanning electron microscopy, because you're going to get a 3D image. So again, similar to that of electron microscopy, the principle is you have an electron gun, you have a condenser lens over here, and then what is going to happen is, how is it different from your transmission electron microscope? By the image from the electron, which is emitted from the object surface, is going to be emitted from the object surface rather from the transmitted electron microscope. So again, you have an electron gun, and this finally produces the beam of electrons. This is known as a primary electron beam over here, and then this is going to pass through, and finally the electromagnetic lens that is present, and you're able to rapidly scan the surface of the specimen completely. So when the beam of electron strikes a specimen, secondary electrons are released. This is going to be transmitted to the electron collector. Once the the electrons, secondary electrons are transmitted to the electron collector, this is it is used to generate a signal, and this produces an image on the cathode screen. So resolution is around 50 angstroms. How are you going to see the resolution of the magnification? So the magnification is the scanned lamp on the screen that you're going to get by the scanned lamp on the specimen surface. Where is it applied? Again, many places. You can see all the small surfaces, specimens, the tissues and the membranes completely. So where are you going to see the applications of microscope? Medical microbiology, all known, detecting pathogenic bacteria, various types of microorganisms, motility, and the movement of cells also can be seen. And how are you, sorry, shows the differentiation between the internal structure, especially for if you want to see cilia, flagella, all of it can be seen. And then use of electron microscopic structures smaller than zero point 0.2 micrometers can be resolved. And fluorescence can help in distinguishing between different kinds of cells. So if the some microorganisms are small, so to properly study the different microorganisms, to culture them, and for that, you need proper culturing methods also, so you will be able to see a better image of the microorganisms. And so this is all about the basic microscopy. In fact, it was the microscope actually that helped us know the insides of almost all the cells as of today. So this is about microscope. You can just see, I think you have these books with you in your library as well. You can just have a look at it. So now we are done with microscopy. So wait, I will start with cell sorting and fluorescence, right? We have that in the next chapter, fractionation of cells, isolation of cell organelles. That is by flow cytometry and cell organelle destruction by centrifugation. Let me just share the screen with you. So before we start with, what do you call it, right, we need to know cell disruption as such. So we will talk about cell fractionation, but before that, we will start with cell disruption methods. What exactly is cell disruption? So you need to know the different kind. You cannot take the whole tissues to see what exactly is there in it. So you need to use cell disruption, that is extract the cell, the desired product out of the cell for us to actually see what is necessary. Yes, there is going to be a physical damage, but still we need certain, if you want some enzymes out of it, or if you want some cell organelles. So all of this to be taken out, you need cell disruption methods. So it is necessary for extraction, retrieval of the desired products, because it will help in the recovery of whatever desired product that we have. So there are several types of cell destruction methods, which is it is also known as cell lysis also, wherein what is happening is the extracellular or the plasma membrane is going to be disrupted. Intracellular, yes, you can extract the organelles that are present, or you can use a periplasmic method of disruption of the cells. So again, they are of two types, mechanical and non-mechanical. And then you have, based on the because different cells have different structures. Your epithelial cells are different from your columnar cells. Simple. And in the tissue, liver tissue is going to be different from your muscular tissue or your brain tissue. So because of which you need different kinds of methods to extract the contents. So what are the cell lysis methods or cell disruption methods? You have mechanical, non-mechanical. Mechanical is you have a homogenizer. You're going to take an isotonic, buffer, you can be trisacetate, trisborate buffer, whatever for that matter, TBA, TBE. So, and then you have a bead mill. You can add some silica beads to it or glass beads and then disrupt it. You can use even a mortar pestle for basic crude extraction of cell contents. Non-mechanical is you have physical, chemical, and biological methods, wherein physical is by heating. Even when you heat, we know that enzymatic activity reaches its peak finally after that the cell lysis is taking place. Osmotic shock, cavitation is one more thing. And then you have in chemical methods uses of alkali and detergents. So you have your tritonics, you have different kinds of detergents which are actually going to what do you call it, break the protein. So because of which you can the cell contents are going to come out. Enzymatic in the case of biological. So we will just have a look at it. Why is it that we are seeing? Because we have to, whatever product that we are looking at after you're disrupting the cells, that has to be released to the maximum. There should not be any kind of denaturation of the product during disruption. That is very important. So it should not be mechanical or heat induced. And then you should have a minimal release of proteases. So you know that anything is made up of protein maximum. So in case there are any proteases released, it will cause damage to the product. And then whenever there is any kind of other particles also or contaminants that is there, it should not actually influence or contaminate your downstream processing. That is your final result. So, and then you're going to also see the susceptibility of the cells to the disruption. How good are they, or how better are you going to disrupt the cells? And is the product going to be stable? How easy is it for us to extract the product from the cell debris? And how fast is the method and how cost effective it is? So what you have to do is cells should be separated from the culture medium. And then once this is done, the extracted components have to be separated and decreased, and all the unutilized media what is there, that has to be reduced. And then cell mass should be dried so that you do not have, it should be cost effective as such. So when it comes to the mechanical methods, you're going to submit it or subject it to very high pressure, or you're going to use abrasion with speed agitation, beads, or ultra. So you have a method called as sonication. So methods of cell disruption include the cavitation, shearing, impingement, or you can use all of them combined together. Intensive cooling is necessary because all of it involves generation of heat. So you can keep it in an ice pack, or you can put it in an ice bowl so that heat is going to be like whatever heat is generated is going to be come. So usually whenever you do any kind of cell disruption, and before that you're going to do homogenization, that or you're going to use beads or any of this, you're going to do it in ice. That is what we usually do in labs. And you can use high pressure methods such as the French press or the Humanis press. So basic is your pressure, because of which the cells are going to be disrupted. So when there is a bead mill, you have beads that are there. So you have the cells that are there. So in between the beads, it's a roller. It is going to be rolled like this. In between the beads, the cells are going to get crushed, disrupted. You can use agitated also. You have the agitator and then small beads, and then they collide with each other in the chamber. So you have the glass ballotini or stainless steel balls that are used, depends upon the different sizes. And then you can increase the number of speed if at all you want to increase the degree of disruption. So because of which B2B interaction increases and your disruption is much more effective. But heat and power is going to be consumed. So this is the liquid phase product that is present. These are the beads that are present. If the beads are too small, you're not going to get what do you call it, the complete product. And if the beads are too large, it is going to be product particle is going to be small, and you're not. So based on this, you have to select the bead size, number of beads. So what are the variables? Agitator speed, proportion that is present over here, and bead size, cell suspension concentration, cell suspension flow rate, agitator disk design. Kinetics include that, with the time and speed, they are going to make a difference. So what is the protein content present in the cells? Time is the amount of time required, by the particle to spend in this, bead mill. And then K is the release constant dependent upon the system. And PM is the maximum possible protein releasable. So again, what are the disadvantages? With the increase in the temperature, so bead volume is also going to increase the bead volume. So cooling is required, and you cannot exactly use large quantities, and high chances of contamination is always there. Ultrasound, this is your sonicator that is being used over here. So ultrasonic disruption, what is going to happen is vibrators are used, high frequency sounds are present. So wave intensity is around 20 kilohertz. So transducer is present, converts the waves into mechanical oscillations. This is the titanium probe. You have an ice bath over here. This is the cell test tube sample. So it is going to be useful when the sample is very small. So what is going to happen is, this is the bubble that is being there, initial collapse because of the sonicator and the high frequency sound wave that is being produced. So jetting takes place, release of the particles. So you use it only for very small, samples that are present, and has high energy requirements. And health and safety issues are there basically because of the sound that is being produced, and it is not going to be continuous. You cannot go on doing it. You have to really stop and then again proceed with it. So this is the drawback that is present in the ultrasonicator or ultrasound frequency disruptor. French press, basic, something like your roti press that you have. High pressure homogenization, cell suspension is done through you have a plunger over here. This is the cell suspension, and then you're going to add pressure to it. This is the orifice and the jet, and this is the impact plate, and you're going to get. So you can use a pressure of up to 1500 bar, and through a narrow annular gap and a discharge valve, pressure drops again over here to the atmospheric pressure. So cell disruption is achieved because sudden drop in the pressure is present upon the discharge. This causes the cells to explode. So when you're using this at higher pressures, what is happening is you are going to get the number of passes of the slurry through it can be decreased. So you're going to get the required amount of disruption. But if at all you're using this whenever there is high pressure, certain heat sensitive proteins will be deactivated. So this may increase the number of passages required through. So you can't use it at once and get your cells to be disrupted. You have to do n number of times to get the all the cells to be disrupted. So what are the factors that are influencing the protein to be released out of the cell, if at all you want protein as a product? Temperature, then the intracellular location of the enzymes, the number of passes, the operating pressure, and the biomass concentration that is present in the cell. So, and then we have homogenization. So, in this again, high pressure homogenizers are there. Your normal lab homogenizers are there, wherein you have that homogenizer, you have the isotonic, you have the buffer. You take a small tissue sample, and then with the ice bath present, because all the time heat should be dissipated. You're going to put the ice bath, and then you're going to homogenize the cells. So you have a displacement pump over here. This is the feed. This is the impactor ring that is present, and this is the homogenized product that is going to be there. So you're going to use a pressure and a pump cylinder. So you have one factor, that is the pressure applied and the pressure drop. because of which the cells are going to burst. So if the pressure is more efficient is your cell destruction. But you have multiple passes, this decreases the productivity rate, and then you get fine debris. Because along with your product, there are other products are also present, which is going to be difficult for us to remove through a further filtering processes. So you can use only one particular pressure which is compatible with your product, so that you can safely the equipment also tends to get damaged, because you need to see the temperature also present. So you have one particular pressure that can be used, and at the same time, that the kind of heat that is going to be dissipated, you will have an idea of it. So you're going to be prepared with whatever sample, different kind of heat dissipators, water bath, ice bath, whatever. And then see that your enzyme also should be stable at that particular temperature. So all of this has to be taken into consideration when you're using a high pressure homogenizer. And then the valve unit, whatever is there, is going to be eroded. So that has to be maintained because we are operating under pressure. Now, when it comes to non-mechanical physical methods, so you have thermolysis. So what are you going to do? Basic heating up. So what are you going to do over here? For example, in gram-negative bacteria, periplasmic proteins are present. They're going to be released when you are going to heat the cells up to 50 degrees Celsius. Cytoplasmic proteins, 10 minutes if you heat at around 90 degrees Celsius, they're going to come out or released. And then, so heat shock. So you give maximum heat for a little while, and then because of which you have more amount of protein that is going to be released. Then when you do it for a longer time at a lower temperature or longer temperature, then what is going to happen is you're not going to get a good amount of protein product being released. So this has to be taken into consideration. So the results are going to be unreliable, because what is going to happen is, if at all there is a temperature fluctuation, your protein solubility is going to change. So not exactly a very feasible method. You also have one more second method, which is known as the decompression. In this, what is going to happen is, you have a gas that is being mixed with your cell suspension. It is going to be under pressure for a specified time and for specified cells. So this gas is going to enter the cells. Ions can diffuse into the cells, expands or release. This causes the cells to burst. So how is it advantageous? So you have carbon dioxide which is used. This is going to be used to extract the flavor, the liquid components, because CO2 and different gas ions can enter the cells, and it is gentle on the cells. So you have large amount of debris that is going to be easier for you to remove, so that only a desired product is formed or product is obtained. But you have a disadvantage in this, which is it is not very efficient. It is dependent upon the amount of pressure that you're putting on and the amount of contact, or between the gas and the cell. So this impacts. So if you don't expose the cells to gas for a considerable amount of time, then the decompression is not going to work efficiently. You have osmotic shock, wherein you're going to add either hypotonic or isotonic or hypertonic solution. So if you take cells, put them in salt concentration, what is going to happen? Cell lysis is going to occur. So this is what we are talking about in osmotic shock. So cells are exposed to either very high or very low salt concentration. So because of which water is going to come out, cell is either going to burst or cell is going to be lysed. So based on this, you're going to get the desired cell product. But again, it is not very efficient. It needs a pre-treatment of the enzymes so that the cells are weakened, and then addition of high amount of salt is necessary. Again, you need to use high amount of water, with which your product can either be diluted, concentrated, or changed as such. So your downstream purification process is going to be very cost inducive. So these are the disadvantages. Now, when it comes to non-mechanical chemical methods, you're going to use different kinds of detergents. What is going to happen using detergents? The lipoprotein on your plasma membrane or any microbial cell membrane is going to be damaged or cut. So these detergents are again divided into anionic, cationic, non-ionic detergents. Most commonly used, we have used it many times, sodium dodecyl sulfate, Tritonix 100. You also have your ethyl trimethyl ammonium bromide, which is a cationic detergent. So based on the ionic nature, that is like, your anion or cation, you're going to have these detergents which work on the different ends of the protein. So the amino end or the phosphate end, it can work at different ends. So STS page is what we usually use. So for your protein, separation and analysis. So sodium dodecyl sulfate reorganizes the cell membrane. So what is it going to do? Disrupt your protein interactions. Tritonex for membrane proteins again, cationic detergent, that is your ethyl trimethyl ammonium bromide. It acts on your LPS, that is your lipopolysaccharides and your phospholipids. So what is it going to be done over here? You can just see they interact with the cell membrane. Then this causes it to break down. So the cell membrane is going to be lysed. The DNA is going to come out. So, but the disadvantages, your proteins are denatured. Detergents also disturb the downstream processing steps. Again, you need purification that is present. You can use solvents also, such as your alcohols, dimethyl sulfoxide, that is your DMSO, it's known as methyl ethyl ketone, toluene. So, with these solvents are usually used to extract the lipids. So, again, this leads to the release of all the intracellular components. You can use alkali soaps. You alkali treatment. So again in this, what is it used for? Hydrolysis of the microbial cell wall material. So desired the enzyme will come out, will tolerate, this is around pH of only 10.5 to 12.5. This is for around half an hour maximum. But then again, you have a disadvantage. Neutralization of the alkali is going to be there. Product need not be stable in alkali conditions. You have non-mechanical enzymatic methods also. So you're going to use enzymes to disrupt the microbial cell walls or the cell wall. So we are talking about bacteria over here and the microbes. So you can use lysozyme. So lysozyme is enzyme. Lysosomes also have it, right? So that is present in all our cells. So they are the ones which are used to. Again, you can use a specific enzyme based on the specific microbe. So lysozyme is commonly used. Will digest the cell wall of the gram-positive bacteria. Where is it going to act? On the beta-1,4 glycosidic bonds that are present in the peptidoglycan layer. So enzymes are usually used. They are the cellulases, pectinases, xylanases, chitinases. So based on what you're looking at the product, you can use these enzymes. But then for it, you need to use purified enzymes. So it is again going to be expensive. And the amount of enzyme that is available for you to use on the particular, for the product to be obtained is also limited. So in a large scale process, it's difficult to do that. Not impossible, but difficult, and at the same time, very expensive affair. And the added enzyme can also complicate the downstream processing. So that is your purification. Again, you need to do more purification. And then, if at all you're seeing that, you can use the immobilized enzyme so that you can get a purified product as such. So finally, what are you going to see? You have these mechanical methods available at the lab because of the cost effectiveness and scale, you can't upscale it. So mechanical methods for industrial purposes. And what are the disadvantages when you're doing the mechanical methods? You need high energy and high pressure requirements. So again, you have different variables that are playing an important role in the efficacy of the mechanical methods. So ultrasound, if at all you use, it is going to be only for a small amount of product or sample. So difficulty of the sterilization, cleaning also makes it more susceptible to contamination. So sonication also has, that is your ultrasound, it is a mechanical method. It is going to generate a very intense noise even for that short time that you're using. I'm sure some of you must have used sonicators. I do not know whether you have done it or not. It actually produces a kissing sound, and it is very loud. And then so this is not exactly useful or health safe for the health when you're using it for a long time, prolonged time. And then mechanical physical methods have prerequisites, so you need to take care of that. At the same time, we should see that the temperature which is going to be released, the heat that is going to be released, does not affect your product. So changing the temperatures used in thermolysis causes the cells to burst also. They may damage the cells. So temperature variation affects the enzymes and the 3D structures. So, and then all of this is expensive. Chemical methods, again, disruption of the sensitive cells. Solvents, what is happening is protein denaturation. So if your final product is going to be a protein, it can also be denatured. There is a chance. So again, removal, recovery of the chemical disruptor is going to be expensive. And then all these chemical methods have only a low efficacy. So they're going to be expensive and less useful as disruption methods. And then high consumption of solvents, water. So not very environmental friendly. Enzymes are gentler, but very expensive again. So this is all about the cell disruption methods. Now I will talk about the flow cytometry that is present. Give me a minute, I will just start with flow. After this, we have the tracer technique, and then cell disruption methods is over, right? So I have flow cytometry. Screen sharing is progress, right? Then after this, I will be dealing with the tracer techniques also. So, can I have a phone? 1 minute. What? Yeah, we were dealing with cell fractionation also, no? Like I will just have a look at it, like share the screen. Yes. After this, I will be talking about tracer technique. Oh, the remaining chapters, whatever they that are there, I will deal with once the time is 1:15. I will talk about them in the next class.
Okay, ma'am.
So give me a minute. Right. Is it one? Right. Share screen. Share. Right. So when it comes to cell fractionation, what are we seeing basically over here? We are going to separate the particles that are present in the cell, and based on the please give me a moment. I'm at that particular slide that I wanted to share with you. Right. So right now we have flow cytometry, disruption of the cells that we have already seen, organelle cell separation by centrifugation, antibody specific used for purification of cell organelles. So that is there. We have tracer techniques and auto audiography. So today I will be able to deal with this fractionation of cells, that is your flow cytometry. Already I have dealt with the disruption of cells. And wait, my camera is off.
I'm sorry to disturb.
Yeah, tell me.
It's it's not visible, ma'am. That's why.
No. Yeah, that's what. Let me just share once again what happened to it. I was seeing. So I'm talking to you as such. So today I will talk about tracer techniques. We have 2.6, which is your audiography. What are the different kinds of isotopes that are being used in the emulsion and the choice of emulsion and film? This I will deal with in the next class. Mostly we will not have time after tracer techniques. If at all there is time, I will try to tell. Okay. Now I will talk about flow cytometry. We have already spoken about, just now I finished different kinds of disruption of the cells, then the organelle separation by centrifugation. I will just talk about it. So now I will start with flow cytometry. Right, let me share screen. Right. So what is it that we're looking at in flow cytometry? It's been there for quite some time. So right since, what do you call it, AIDS and all that, yeah, groups, someone add me to that first year group, no, then I will send whatever I'm showing you on the screen. I will send all these PPTs. I have taken them from the internet, so as it is, I'm going to just send them to you. So anyway, when it comes to so just add me to your MSc zoology group that is there, and I will just share them, yesterday's and today's. So back to flow cytometry. So whatever this immunophenotyping was seen right since the time of your AIDS. So 1983 onwards, 1980s onwards. So what was it used to see or different assess your CD4 T cells, lymphoid cells. Now myeloid neoplasm, that is your cancer cells, all of it are being seen. So you can simultaneously measure different cells in from the single cell suspension that is there in your flow cytometry. Large number of cells specimens can also be processed, and it is quite sensitive, so you can detect good types of cells and different types of phenotypes also in the cells, and the specimen number can be thousands. What is it that we are going to consider when we start with flow cytometry? Size of the cell, and what is the kind of cytoplasm granule, that is your cytoplasmic granularity that is present, the density, the amount of DNA present in the cell. And you're going to see whether the cells are the antigens that are present, what are the different kinds of markers, so that you can mark them accordingly. So are we looking at the surface markers, or are we looking at the cytoplasmic markers, or are we looking at the nuclear markers? So that this is going to be the antigen, and they can be recognized by the specific antibody, and there is a fluorescent tag attached to it. So what is it that we are going to do? Or whatever is it that we are going to learn through flow cytometry? What is the kind of cell that is going to come out? Is it hematopoietic, or is it lymphoid, or is it non-hematopoietic? That is your lymphoid. And what are the kind of cells that are coming out? Are they B cells? Are they T cells? Or are they new NK cells? Or are they myeloid? Or are they endocrine cells? Or are they epithelial cells? And then what is the maturation of the cell? Are they precursors that are coming out, or are they the matured B cells or the matured killer cells that are coming out? So what is the principle of flow cytometry? So optical and fluorescence characteristics as such. So you have a fluorescent tag that is attached. So in that particular cell, is there any antigen that can be tagged using a fluorescent tag as such? So during this,
Hyderabad group page.
So during their passage within a narrow,
Right of
precise,
Bagista
defined
Bachmara.
liquid. I request you to mute your microphones. For cell analysis, what are you going to see? That is your flow system, optical system, electronic, and then computer. And then you can save the images also. So what is going to happen in your flow cytometer? Flow cytometer. These are the cells. This is the cell sample. You have the nozzle over here. Looks very simple, no? Cells are going to come out. There's a laser beam attached over here. This is the cells again. So you have a laser beam. This is going to do all the work over here. Once this is there, it is going to, you have an obscuration bar. Blocks the light over here. So it goes through to the dichroic mirror. Over here, you have different sets of dichroic mirrors. And then you have the fluorescent tags over here. So you have different kinds of filters attached. And this is going to be attached to your analysis computer. So what are these PMT tubes? Your photomultiplier tubes. And then you have the analog to digital converter that is present. Right. Now what is going to happen over here? Physically, size, you're going to have a scatter of the light because of the size particle. So you're going to have the forward, the slide scatter and the forward scatter. So what is going to happen is, according to the light source that is present, the angle, so you have a side scatter detector over here and the forward scatter detector over here. So internal complexity can be seen. So this is going to resolve you're going to get a better resolution. So it collects the light at around 180 degrees from the point at which the laser beam intersects. So it is going to, the image is on the linear scale. Again, correlation with the cell size is taking place. Then because of which you're able to distinguish between the different types of cells. Are they normal cells? Are they monocytes? Or are they cancerous cells? That is your neoplastic cells, which are usually going to have different cell surface, different cell size. They are larger in size. So the cell surface collector collects the right angle light at 90 degrees. This is again going to correlate with the cytoplasmic granularity. So because of which what is going to happen is you're going to get the cytoplasmic image as well and the nuclear configuration. So you're going to combine both the images and then you're going to distinguish between the different types of cells present. So this is how the end image is going to come. So if it is a neutrophil, you're going to get an image as such. If it is a monocyte and if it is a lymphocyte, based on the kind of scattering of the lights. That is why the laser beam makes an important plays an important role along with your dichroic mirrors that are present. And all of it will help in getting the correct image of the different kind of cells that are present. So when you're doing immunophenotypic analysis, you're going to get antibodies and fluorochromes. So monoclonal antibodies are used, and all of this are based on the cluster of differentiation, that is your CD. So identification and distinction of the cell surface antigens. Is it okay if I take around 5 minutes of your time and try to finish tracer techniques today?
Yes, ma'am. No issues.
Okay.
Yes, ma'am.
So, all right then. Yeah, thank you so much. I'll try to finish tracer techniques today. So this is again highly specific antibodies are used, monoclonal antibodies etc. They are used to identify. So you're going to you have the surface markers to which the antibodies are going to be attached. So you have a fluorochrome. So this is going to change after upon the light beam and the wavelength change, because of which you're going to get fluorescence. So when this fluorochrome is attached to the antibody, which is attached to the antigen or the cell surface marker such as CD4, CD8, CD3, etc., you're going to get fluorescence over here. So fluorescent dyes can directly bind to certain cellular components, your DNA, RNA that is there. So you're going to get a quantitative analysis of that particular individual cell. This can be conjugated with the monoclonal antibodies. So you're going to get a target cellular antigen or marker. So what are the fluorochromes that are usually used in your flow cytometry? That is the fluorescin, phycoerythrin, then you have your L-phycocyanin, etc. And you have the excitation wavelength and the emission wavelength. At what wavelength you can see, or at what wavelength it is going to be emitted. So some of them, if you see same wavelength, they can be excited at the same light source, but your emission wavelength is differing over here. So according to that, you need the emission filter, which is different. So for example, you can use both FITC and PE with same excitation wavelength, so that filter can be same. But emission wavelength, if you put two different filters, you can get both wavelengths. And so at the same time, you can see a blue and a red color. For example, I'm just like telling you. So you can see fluorescent isothiocyanide, and at the same time, you can see phycoerythrin together in the same cell. So this can be simultaneously multiple fluorochromes can be excited by a light source detected by the emission fluorescent signal with different wavelengths respectively. Phenotyping analysis, what is happening? This is the sample. These are labeled, and then through the flow cytometer, you can get the analysis and the product can be seen because you're using two different filters over here. Simple. So simultaneous detection of the multiple cell antigens or markers is taking place. Like I told you, you can see FITC-PE at excitation at one time, emission different. So accordingly, fluorochrome conjugated specific antibodies are present over here. Different fluorochromes have different emission wavelengths. They can be simultaneously detected by the flow cytometer. So what is it that you're going to see? Abnormal or aberrant antigenic expression can be grouped into four basic categories. So what is it? Aberrant expression. Gain of antigens not normally expressed in the cell type, they can be seen. Expression of the antigens that are not synchronized with normal development or maturation of the cell type. This can be also seen, and homogeneous expression of the antigens by a cell population can also be seen. So how is it that is going to be seen? You just see CD4 antigen markers are present, CD8. So helper T cells, killer T cells, both of them are there in the sample passed on. You have the detector over here. The data is collected. You're going to be seeing them. This is the result that you're going to get and analyze. So how are you going to process the specimen for the flow cytometry? So lack of distinct antigens or markers can be seen basically. What is it you're going to take as a sample? Blood sample, bone marrow, body fluids, CSF, urine, all of this can be taken. So you have fine needle aspirates also. If there's any kind of pus cells that is there, you're just going to take using the fine needle aspirates. Store them appropriately using different kinds of anticoagulants or storage substrates such as EDTA or citric acid dextrose. Fresh specimens should be transported properly, stored in sterile conditions, and then you can use refrigeration in case there is any kind of delay in the cytometric analysis, and then you can use this. So for this, what are you going to do? Single cell suspension is best used. Then the narrow bones or of the sample injection needle tubing can be clogged by aggregated cell debris. So you have to take care of that. Make a suspension. And if it is a solid tissue, according to that, mechanical disruption is done. You can use enzymes, enzymatic digestion, mechanical disaggregation, centrifugation, basically. So finally, you're going to get the cell suspension, which is seen through attached with required antibodies, and then seen analyzed through the flow cytometer. Applications, you can see a DNA content analysis. You can use dyes such as propydium iodide. You can see the DNA helical structure. And then if there is any loss or gain in the DNA, telomerase loss or DNA content aneuploidy, tumor cell population, all of this can be seen in flow cytometry. So different phases are there. Different cell cycle phases can also be seen in the flow cytometry. So this is the kind of DNA that you're going to get. Erythrocyte analysis can also be done, especially fetal red cells in the maternal blood. So if there is any kind of a problem in the fetal red blood cells as compared to the maternal red blood cells, then you can actually see that. And diagnosis of PNH, which is a disease, and you're going to use what do you call it, the flow cytometer. If there's any kind of deficiency, then you're going to find out. Reticulocytes, they can be analyzed, and blood bank also you can use it for different kinds of what do you call it, recipients and phenotype of the donor and recipient basically. And if there's any kind of leukocyte contamination, then you can see that. So analysis, all of this can be done or detected using your flow cytometer. You can find out different kind of cancer cells, all your B cell lymphomas, whatever is there. We're using flow cytometer analysis. Our Vijaya Diagnostic Center is very well known for detection of different cancers because of an amazing flow cytometric analysis that is present with them. So, if, depending upon the markers, you can find out different kinds of leukemias that is present. Identification of lineage-specific antigens and immunologic monitoring of HIV infected patients. So, like I told you, flow cytometry came to existence in 1980s, wherein CD4, CD8 markers, T cells, helper T cells were used to be identified. So basically, the HIV viral load and the CD4 T lymphocytes were used. Are you? You're still able to see the screen, right? Just a second. Right. So lymphomas, myelomas, leukemias, all of it can be identified. Platelet analysis can be done. I will send this particular I will share this particular slide with you. It is very good. Interpretation is also seen over here. So you have the forward light scatter and the side light scatter, through which you will be able to find out if at all they are the size, granulocytes and the monocytes and the lymphocytes. So you already know the sizes over here, the reference. So with this analysis, you can make out as to what kind of cells they are and if there is any kind of anomaly inside the cells. So to determine whether the cells are hematopoietic, you can know after expression with CD45 antigen, then you're going to see the size of it. And according to that, they are divided into CD45 plus, CD45 minus. Then they can be separated based on the histogram that is going to come. So are they plasma cells? So are they CD45 negative or positive cells? So according to this, you are going to see whether there is any kind of abnormality present. So more CD45 positivity, more brightness is seen, and whereas the others will have a lesser brightness. So finally, what is going to happen is it is going to be gating, wherein first step of phenotyping, immunophenotyping analysis. And in this, the specimen is mixed, and then you're going to get a bone marrow aspirate. So you're going to get different types. On there is going to be an upper and a lower limit on the kind of amount of specimen that you're going to pass through. Then again, you can divide it according to the subpopulation from this heterogeneous population that is present, and then get a more specific type. So what is the type of gating? Cell distribution into CD45 versus side scatter and cell size. So you're going to have an FSC and SSC, that is your forward light scatter and side light scatter. According to that, you're going to get an analysis. And at the same time, you can see if at all there is any neoplastic cells, you're going to get what do you call it, a bigger cell size. So end of the day, with your fluorescent light microscope, flow cytometry, you're going to analyze the sizes and according to that, distinguish the different types of cells that are present if there is any kind of abnormality. So size control is one thing. And then cell lineage specific antigens. So immunophenotypic is done. So, and then gating of the lymphocytes, once this is done, it is going to be around the lymphocytes. You're going to get one particular lymphocytes are colored in red. So the rest is again going to be of a different type. So where are they present based on the size? And you have the FITC conjugate CD8 cells, FITC conjugate and then you have the lymphocytes. The monocytes are going to be in. So C is a gate that is present, and it has been set only to show the cells in the R2. So, and then if at all you see T lymphocytes, if you want to distinguish between them, so you're going to use a different kind of peak. You're going to get lymphocytes over here, T cells over here, and then you have a C20 FITC conjugate. So you have, this is all the analysis of your flow cytometer. So again, quality control, you have quite a lot of quality control that you have to do over here. So you have fluorescent beads that are used for quality control. Second quality control is going to be the setting of the instrument, how you're going to do, what is the kind of stains, beads or antibody stain cells. Third is the level of quality control or the control substance that's being used. So stabilized blood, and you have added tissue culture cells that are going to look like your if you're looking for or you're suspecting a specific cancer, then you're going to use a tissue culture specific cell so that it mimics that cancer cell. So when you're seeing different immunophenotypic techniques, you have flow cytometry and immunohistochemistry. So shorter time, but histochemistry is going to take. Histochemistry is preparation of slide and looking at into your using a microscope as such. Whereas in flow cytometer, you can detect it quite quickly within hours. So result interpretation is also going to be quick, because you know what kind of cancer is already there. You with your different specific markers that are being used. Over here, you need to analyze and then come up with, okay, it can be a different kind. If it can be leukemia, but over here, you know what specific leukemia it can be, because you have your CD, your lymphocyte analysis that is being done quickly with the different kinds of markers that you're going to use cocktail markers and come up with a result immediately. So you can use multiple antibodies. And then again, you cannot do the same thing over here when it comes to immunohistochemistry. So non-viable cells cannot be assessed, but you can assess the non-viable ghost cells or the cells that are also there. So you have the differences when you compare different kinds of immunophenotypic techniques. Flow cytometry is necessary, and it is a very good qualitative, quantitative technique when it comes to your analysis or identification. And then it is smaller. You don't have a lot of except for your what do you call it, installation costs. So you can use it. You can use different kinds of antibodies for routine clinical laboratory, especially for your diagnostic centers. Flow cytometer is very useful. So disease diagnosis, management of the disease also, you can use flow cytometry. So it is quite advanced and it is quite good. Unfortunately, I just could complete this. If you still want me to start with tracer techniques, I will do that. This is around some 14 slides. So I'll try to finish it off. It's quite possible. Are you able to see the screen? In
Yes,
another 5-10 minutes, I can finish the tracer techniques. Okay. So where exactly are tracer techniques used? So when it comes to that, you want to see living plants, and you want to see the metabolites that are present in the tracer techniques using tracer techniques. So you're going to use radioactive isotopes to trace the pathway. So what is it that you're going to find out? You want to know about different kinds of secondary metabolites that are present. Usually in the plants it is used. So if you want to differentiate or know the biosynthetic pathway of the plant metabolites, for example, if it is shikimic or mevalonic or any kind of pathway. So you have a lot of intermediate steps that are involved in this. So how is it that you're going to find out all of these pathways? So you use tracer techniques. So or else you can isolate that particular organ and find out, or you can use grafting methods or mutant strains. So what is the basic definition in the tracer technique? You use a radioactive labeled compound to find out the different intermediates and the steps in the biosynthetic pathways at a given point of time at a given rate. So usually you use different isotopes, radioactive isotopes that are incorporated into the precursor, presumed. We do not know whether it is the same one or not, but we still presume that it is the one and then incorporate into it in for finding out the different secondary metabolites. So it's a marker as such. So what are the labeled compounds that are you going to use? Either radioactive isotopes, which can be C14. C12 is your normal isotope, but C14 is the one. Some slides are not changed. Oh, you're not able to see the slides? Wait, I'll pause. You share. I think now you can see. Resume shares. Yes. So definition is, you have already heard the definition. So what are the different kinds of radioactive isotopes? Tritium is one, hydrogen is one, C14, P35, then sodium 24. These are all the different kinds of isotopes that I use. Usually you use C14 for biological investigations, and then hydrogen, tritium ion, H13. So for metabolic studies, you use sulfur, phosphorus, and different kinds of alkali, alkaline earth metals that can be used. You use even stable isotopes. What are they? They are going to be labeled as possible intermediates in the biosynthetic pathways. So you can distinguish them or find out them using mass spec or NMR spectroscopy, depending upon whatever isotope you're using. How is it going to be useful for us as such? So if at all you're incorporating your C14 into the phenylalanine, so what is going to happen is you're going to find out the cyanogenetic glycoside pronacin. It can be detected if at all it is present or not in that particular pathway. Location and quantity of the compound containing tracer is also seen. So if at all you radioactively label the glucose with carbon 14, then you can find out how much amount of glucose is there in that particular system, life system. Different tracers are used for different studies. So because of which you can get the amount of or determination of the amount of that particular nitrogen or amino acid in the system. So what is it that we're looking at when we are going to use a tracer technique? Whether the concentration of the state tracer, whatever amount we are incorporating, is able to withstand the amount of dilution in the system when the metabolism is taking place. If it is going to disintegrate and degrade, there's no point in using the tracer altogether. And then you have to properly label the physical and the chemical nature of the compound. And then this should involve in the synthesis reaction also, because whenever that labeled compound is used, it should get incorporated and it should be in that metabolism, or else if it comes out as a byproduct, there is no point in using that particular system altogether, that particular tracer. And it should not damage the system when it is used. Advantages, it is highly sensitive, applicable to all the living organisms. You have many isotopes that can be used. It is a very reliable and easy procedure to be done, and you can get accurate results. What are the limitations? Of course, kinetic, chemical, radiation, high concentration, if at all it is going to damage, that is going to be seen. What are the requirements for a tracer technique to be done? Labeling of the compound, that is, you have to preparation of the labeled compound, then introducing that compound into the biological system. Then you have to separate and determine the labeled compound in the different kinds of biochemical fractions that you're going to get. So you can label the compound by, for example, by growing chlorella in an atmosphere of C14. Then all the carbon compounds are going to be labeled with C14 isotope. Then the tritium labeled compounds, they are usually available commercially and affected by the catalytic exchange. Another 5 minutes, the class will be over. Please bear with me. So tritium is basically a beta emitter gas. So low intensity radiation energy is lower than that of the carbon. So what is it going to happen? So you have this magnesium, bromide over here with carbon C14. So you're going to get this particular acetate, what do you call it, with MgBr attached, and your labeled carbon is over here with water. So you're going to get an acetic acid which is labeled with C14, and you get MgOHBr as the what do you call it, byproduct. So this labeled compound should be introduced into the system. So you have different methods: root feeding, stem, indirect, infiltration, floating, spray technique. I have a better what do you call it, a PowerPoint, which I will share with you. All of this is again given in detail. How are you going to separate or detect the compound? Using your Geiger counter, liquid scintillation method, gas ionization, Bernstein mass spectroscopy, NMR, or electrometer, then autoradiography. All of these methods can be used. So finally, what are the methods in the tracer technique? You have a precursor product sequence. So presumed precursor under investigation. Okay, you assume that this is the C14 that is going to be fed into it. Again, what is the disadvantage is that it is not usually enough evidence that this particular compound is the direct precursor as such. So that is one thing you are speculating over here. Okay, next class will start, it seems. So I have around 4 more slides. I'll just try to finish it off in 2 minutes. So where is it going to be applied? This precursor product sequence in barley seedling production and distinct from the hyoscyamine. So this is applied for biogenesis of morphine and alkaloids. Double and multiple labeling, you can use 2 isotopes basically. Finally, where is it applied? For biogenesis of secondary metabolites. Competitive feeding is used for again propane alkaloids, distinguish finding them out. So you're going to use the alkaloids using C14 labeled compounds. So normal route is not the subsidiary pathway. If that is the case, you're going to get a product A gives rise to C, labeled A, B, A to B, B to C, etc. Isotope incorporation method gives information about the bond cleavage and what is the kind of bond that is formed. So glucose 1-phosphatase cleavage catalyzed by alkaline phosphatase, if at all you label, so you can see where exactly the bond is being cleaved. And finally, sequential analysis. So every particular sequence, you can see exposure for 5 minutes with C14 isotope. You can find out the sequential analysis of menthol from in the plant Mentha piperata. So what are the applications of tracer techniques? Study of squalene cyclization. So you use tritium or C14 labeled mevalonic acid. Different kinds of relationship between the sterols. So you're going to use C14 labeled acetate. Terpenoid biosynthesis. So all your secondary metabolites you're going to use. Study of formation of scopolamine. Then tracer for uptake of calcium. Adding ammonium phosphate labeled with P32, you're going to get uptake of the phosphorus in the plants. How much of it is being going into the branches, leaves, etc. So I will stop my share now. I'm done with the class. Whatever is remaining, I will deal with it in the next class. Okay.